Hi, folks. Welcome back. Today, I want to discuss one more function of molecular biology, and I'll call it the recognition and repair of damage and errors. And I'm going to start this video with our old friend, the slide showing the complete version of the dogma of molecular biology, the one the evolutionists don't want you to see because it's impossible to explain scientifically. And in this slide, once again, we see that the process of transcription creates RNA from DNA, and the process of translation creates proteins from RNA, and the process of replication creates more DNA from existing DNA. And all these processes become even more amazing when you discover how accurate they are. The error rate of these processes is remarkably low. In addition to the specificity already designed into the proteins and RNA molecules that function in the system, as well as the entire system as a whole, checks and balances have also been designed into the system. The same proteins involved in DNA replication and transcription can also detect and repair errors and damage to the DNA. And they do this along with the help of other proteins. So let me explain this further. DNA can be damaged in several ways. For example, it can be damaged chemically through things like cigarette smoke, asbestos, or free radicals. A free radical, by the way, is an atom or a molecule with unpaired electrons. They often contain oxygen, and that makes them highly reactive with other molecules. But DNA can also be damaged environmentally through things like radiation from sources like UV light. When DNA is damaged, it can cause problems like breakage of the DNA strand, or it can cause the DNA bases to pair improperly. And in cases such as these, many of the same proteins involved with replication and transcription are again recruited to perform their functions within the region of damage. They'll open up the strand, repair the damage, and close it back up again. Similar to repairing DNA damage, the processes of replication and transcription have checks and balances built into them that monitor for any errors in base insertion. This is largely through DNA polymerase for replication and RNA polymerase for transcription, along with other editing factors. This process is what is commonly referred to as proofreading. And depending on the family, many DNA polymerases have what's called a 3' to 5' exonuclease activity. And that means that during replication, it can recognize any incorrectly inserted bases, back up one base length, and then excise the incorrect base. It can then reinsert the correct base and then continue on with replication. And during transcription, RNA polymerase II uses a simple proofreading step where it'll slow down over potentially incorrect bases, and if it detects it to be incorrect, it can then remove them. And transcription also utilizes RNA editing factors to help with the detection of incorrectly inserted bases. Although these processes and enzymes are designed for near perfect fidelity, errors do still occur. Errors occur as a result of damage and mistakes in processing. For example, DNA polymerase has an error rate that's been estimated to be 5 times 10 to the minus 7th, which is one base per every 50 million. And RNA polymerase II has an error rate estimated to be at 10 to the minus 4th, which is one base for every 10,000. Now, when errors occur and they go undetected or unresolved, it results in mutations. Mutations are alterations in the normal DNA sequence, and there are many different kinds of mutations. They can be a point mutation, where there's a change that's as small as only one base pair, where one base has been substituted for another. They can be a deletion, where there's a loss of either one base pair, a loss of a section of a gene, a loss of an entire gene or multiple genes, or loss of an entire chromosome. They can also be an insertion, where there's a gain of either one base pair, a gain of a section of DNA, a gain of an entire gene or genes, or the gain of an entire chromosome. Now, most mutations are recessive, meaning they do not dominate. Therefore, they do not have an impact. And remember, as I'm showing here on this slide, we have 2 copies of each chromosome and 2 copies of each gene. So a mutation in one copy doesn't mean a mutation in the other. And only having one copy mutated enables the other one to remain normal. So in this slide, what I'm showing, if we have a point mutation where we have a substitution of a T for a C, as I show in red here, only one of the chromosomes in this case is affected, so the other chromosome continues to have its normal gene and normal gene function. And then when you consider that most genes encode enzymes, there's typically still a high enough transcription rate of the normal gene to compensate for the mutated gene. Now, even though the majority of mutations are recessive, every now and then there are some that are dominant. And there are several reasons why this could be the case. For example, the amount of transcription from the normal gene is no longer able to do its job adequately. Or the mutated gene product interferes with the activity of the normal gene product. And this is the case in some cancers that involve tumor suppressor genes. Or there's a gain of a new function with the mutated gene product. Now, a mutation as small as a point mutation, where we have just one base pair changed or deleted or inserted, can have a surprisingly large effect on the final gene product. Altering one base can change the codon being read during translation, and that'll change the amino acid that's encoded by that codon. It can also change every amino acid downstream by throwing off the reading frame of the reading frame for the ribosomes. So you have one inserted or one deleted, that switches everything by one base pair, and that'll change every amino acid downstream of that. Changing just one base pair can also introduce a new stop codon, causing the protein to be truncated or cut off from that codon from that point forward. Single base changes can also alter the bases that signal where the introns are to be spliced out of the mature mRNA. And if an intron is not spliced out, there'll be a whole bunch of additional amino acids that'll be incorporated into the protein that are not supposed to be there. And if the base change occurs in a regulatory region, like the promoter or an enhancer region, it can affect the rate of transcription of that gene. Larger mutations can cause an entire gene or entire genes, plural, to either be deleted or duplicated. So this will affect the presence or absence of entire gene products or maybe regulatory regions of a gene. There's also things called translocations, where large regions of the DNA will switch places in the genome. Now, mutations are also either somatic or germline. Somatic mutations are those that accumulate during a lifetime in cells that are not involved in the creation of egg or sperm. Hence, that becomes the vast majority of our body and the vast majority of our mutations. But germline mutations are those that are found in the egg and sperm cells. And it's critically important to point out that only germline mutations are passed on to the next generation. Somatic mutations are not. This is the reason that an individual may develop cancer during their lifetime and still be able to have children that don't. If the cancer or any other mutation doesn't affect the development of egg or sperm, it's not going to be passed on to the next generation. Well, whether or not they're passed on to the next generation, sometimes DNA mutations do cause disease. And despite the fact that most mutations are recessive and don't have a detectable impact on the individual or their offspring, sometimes mutations, both large and small, can have a severe and lasting impact on an individual. But the good news is that the mutation rate is very low. Mutations are, in fact, rare events. Mutation rates are typically measured in the rate of nucleotide substitution per base pairs, per gene, or per genome, per generation. And a generation is the doubling time of all the cells in a given population. Now, the mutation rate in humans has been calculated to be approximately 1.1 to 2.5 times 10 to the minus 8th. So that's between 1.1 and 2.5 base mutations per every 100 million. Nachman and Crowell estimated the mutation rate to be about 2.5 times 10 to the minus 8th per base per generation, while Roach et al. Estimated the mutation rate to be about 1.1 times 10 to the minus 8th per site per generation, and they were using data available from whole-genome sequencing. So from studies like these, we can arrive at our conservative estimate for the human mutation rate to be between 1.1 and 2.5 times 10 to the minus 8th. Now, mutation rates will differ between organisms. So I just quoted you the numbers for humans, but if you looked at mice or sheep or whatever, you're going to find different rates for them. And if you look at viruses, you find out that they have the highest rates of mutations. DNA viruses have mutation rates as low as 10 to the minus 6 to 10 to the minus 8th per base per generation. So that's one base mutation for every 1 to 100 million. And RNA viruses have mutation rates between 10 to the minus 3 to 10 to the minus 5 per base per generation. So that's one base mutation for every 1 to 100,000. So from this, we learn that RNA viruses are the fastest mutating entities on the planet, which is why vaccines against RNA viruses are so problematic, and why we have to have a new flu vaccine every year, trying to keep up with the rate of mutations from those viruses. It's also one of the reasons there's so many problems with the COVID vaccines. But that, folks, is a discussion that I'll just save for a future video. Mutation rates differ between differing regions of an organism's genome because there are known hotspots of mutations within a genome. Mutation rates also differ between cell types of an organism, and mutation rates will differ between genders. there are higher mutation rates in males than females simply due to the greater number of germline cell divisions, lending to the creation of sperm as opposed to that of eggs. Therefore, mutation rates are even higher in older males than they are in younger males. Now, as I've gone through this, I realize that once again I've given you a lot of information in just this one video. But I'm doing this to stress how there's a lot involved in the detection and repair of damage and errors in DNA. And all of it is necessary for life to exist. A lack of DNA proofreading, recognition, and repair of damage and errors by these enzymes and all the other proteins that are involved in these processes would be detrimental to DNA, to RNA, to the cell, and to life. So hopefully you can see that all of these molecules are critically important for these processes, and therefore they are critically important for life. Without any of these molecules there, critical parts of the processes won't happen. And without it, DNA would become progressively damaged and error-filled. It would no longer encode the appropriate and essential RNA and proteins. It would no longer be reproducible, and life would cease. So once again, these processes are critically important. Therefore, as I've already explained, all of these molecules have to be here at the same time, functioning together, or life cannot exist. They cannot accumulate gradually over time because they're all needed from day one, which means they simply cannot arise via evolution. And as I reviewed in previous videos, this again is also irreducible complexity to the utmost. And I'm going to stop this video here, but come on back for the next one, because I have much more to say about mutations and mutation rates that shows that evolution is scientifically impossible. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching, and as always, we'll leave the light on for you.